Welcome to Module 1 of Cells, Tissues and Regulation. Topic 1.1 will be an overview of the cell's structure and function. During the course of this video, you can open your workbooks to Module 1 and try and answer as many questions as you can. Please read over the learning objectives carefully before you start. The learning objectives for Topic 1.1 are to describe the major differences between prokaryotes and eukaryotes, the characteristics and components of the eukaryotic cell, and what makes up the cytoplasm. By the end of this video, you should be able to describe the structure and function of all the important components of a eukaryotic cell. For the best outcome, it is advised that you complete your workbook questions during the course of this video by pausing, or soon after finishing the module. Cells are the basic structural and functional unit of life, and as time has passed, cells have evolved immensely. Currently, cells are divided into two different categories: prokaryotes and eukaryotes. Prokaryotes are the first forms of life to appear on Earth and are one of the most successful organisms to ever live. It is believed that the first prokaryotic cell appeared approximately 3.5 billion years ago. Prokaryotes are single-celled organisms, and the major feature of these cells is that they do not have a nucleus. Hence, the DNA is situated in the cytoplasm. They also have a stiff outer cell wall that is made up of peptidoglycans. The most common example for a prokaryotic cell is the bacteria. The cell wall is really important for prokaryotes to sustain life. Most of the currently used antibiotics, such as penicillin, kill bacteria by damaging or disrupting their cell wall. So let's talk about eukaryotes. These are complex organisms and can be either single-celled or multicellular. The presence of a nucleus, which holds the genetic material or DNA inside of it, and mitochondria are the most distinguishing features of a eukaryotic cell. These cells also have an extensive internal membrane and a network of membrane-bound organelles. These cell types are usually much larger in size compared to prokaryotes and have a very diverse and dynamic cytoskeleton. Some of the best examples of unicellular eukaryotes are protozoans like amoeba. We humans are aggregates of trillions of eukaryotic cells. As mentioned previously, there are trillions of cells in our body which vary greatly in shape, size, and function. However, regardless of these differences, all cells have some basic parts that perform common functions. A eukaryotic cell can be divided into three major parts. The nucleus controls cellular activities and holds all the genetic information. The cell membrane, or the plasma membrane, is the outer boundary of the cell, which acts as a selectively permeable barrier and separates the delicate inner metabolic machinery from the outer environment. The cytoplasm holds multiple different structures within it, such as cytosol, which contains dissolved solutes, membrane organelles such as mitochondria, the Endoplasmic Reticulum, the Golgi Apparatus, and lysosomes that perform specific functions such as protein synthesis or ATP production. Inclusions include lipid droplets, glycogen granules, and ribosomes. Finally, protein fibers make up the cytoskeleton and cellular extensions such as cilia and flagella. It is really important for you to understand the basic structure and function of each of these listed components. The plasma membrane is the outermost layer of a cell that acts as a mechanical barrier separating the intracellular components of the cell from its external environment. The plasma membrane is made up of a phospholipid bilayer that has a hydrophilic head that attracts or dissolves in water, and means water-loving, and a hydrophobic tail, which repels water. The plasma membrane is a fluidic and flexible structure. However, the fluidity can depend on various factors, such as temperature, lipid content, and cholesterol content. The plasma membrane is essential for all life, as it isolates the cell from its surrounding environment. It controls the entry and exit of fluid and materials into the cell, and without it, life cannot occur. Some of the major functions of the plasma membrane include selective permeability, by which it can control the entry and exit of substances such as sodium ions, glucose, and proteins; cell signaling, as it holds receptors for signaling molecules that can turn on and off processes in the cell. The plasma membrane is also covered with a layer of glycoproteins and glycolipids, which play a crucial role in cell-to-cell communication, such as cell recognition and intracellular attachment. The cytoplasm is the viscous region between the nucleus and the plasma membrane. It contains organelles, cytosol, a cytoskeleton, and inclusion bodies, such as lipid droplets and glycogen granules. The cytoskeleton is an elaborate network of rods or filaments running through the cytosol. The primary function of a cytoskeleton is to help the cell maintain its shape and internal organization. There are 3 different kinds of filaments in a cytoskeleton, which are made up of different proteins. The microtubules are made up of tubulin, microfilaments are made up of actin, and intermediate filaments are made up of a variety of proteins. These filaments are needed to maintain the cell shape and act as anchoring points for various organelles. They also play a key role in cell division and movement of the cell. Now let's discuss each of these filaments separately. The microtubules are large and hollow structures and are made up of tubulin. The primary function of these microtubules is to provide stability and a structural framework for the organelles, and is also involved in moving them when needed. Microtubules also play a key role in transporting vesicles. Most microtubules spread from a small region in the cytoplasm near the nucleus, called the centrosome. The centrosome acts as a microtubule organizing center in animal cells. During the process of cell division, a pair of microtubule bundles form centrioles to help in the movement of DNA. A pair of microtubule bundles that form centrioles, as mentioned in the previous slide, also form the bases of cilia and flagella. These structures are located on the surface of the cell and are involved in the motility or movement of substances or the cell itself. Cilia are whip-like motile extensions that can be found on a large number of exposed cell surfaces. The cilia beat back and forth, which is known as ciliary action, and helps move substances in one direction across the cell surface, or move the entire cell itself, which is commonly seen in some prokaryotes. Flagella are substantially longer than cilia and have a rotatory motion. In humans, the only cell type that is known to have a flagella are sperm cells. However, they are common in protists such as Euglena, and also occur in some prokaryotes. Take a moment to consider the following question. The actin filaments are the smallest cytoskeletal elements inside the cell, and they help determine the cell shape. Actin and myosin act to control the movement of structures inside the cell. For example, the cytoplasmic streaming or movement of cytoplasm around the cell in plants. Actin filaments also play a major part during cell division by helping in the process called cytokinesis, which is the division of cytoplasm. In some cases, actin can also be responsible for the movement of the organism itself, as seen in amoeba. The intermediate filaments are responsible for providing the structural support, but not movement. There are various types of intermediate filaments depending on the type of protein involved in different cells. For example, these intermediate fibers are called neurofilaments in a neuron and keratin filaments in epithelial cells. The inside of a eukaryotic cell is divided into membrane-bound compartments called organelles that isolate and hold specialized cellular components and materials within them. Hence, the compartmentalization of these organelles has several advantages, as it allows packing of materials within the cell, permits independent metabolic reactions, and isolates dangerous products that can damage or destroy the cell, for example, the digestive enzymes contained within a lysosome. Now let's learn about the organelles within these compartments. The nucleus is the control center of a cell and is bound by a double membrane barrier separated by a fluid-filled space called the nuclear envelope. The outer membrane of the nucleus is studded with ribosomes and is continuous with the rough endoplasmic reticulum. The inner membrane is lined by intermediate filaments that help maintain the shape of the nucleus. The nuclear envelope is also covered by numerous protein channels called nuclear pores. These pores allow and regulate the movement of RNA and proteins in and out of the nucleus. Most cells have one nucleus. However, there are some exceptions. For example, the skeletal muscle cells in our body are multinucleated, while the red cells in our body are anucleated, meaning that they don't contain a nucleus at all. The primary function of the nucleus is to hold genetic material, or DNA, through which it dictates protein synthesis. The nucleolus is a small spherical structure within the nucleus and is responsible for RNA synthesis and ribosome synthesis and assembly. Ribosomes are non-membranous organelles that have a large and a small subunit that fit together and are composed of proteins and a variety of RNAs, called ribosomal RNA. The ribosome population inside the cell are divided into free ribosomes that float freely in the cytosol and membrane-bound ribosomes that are attached to the rough endoplasmic reticulum. Ribosomes synthesize proteins in the cell. The rough endoplasmic reticulum is a network of membrane-bound tubes and sacs that are studded with ribosomes. One of the most important features of the rough endoplasmic reticulum is not only the presence of ribosomes, but also the fact that it is continuous with the nuclear envelope. The major function of the rough endoplasmic reticulum is to fold and process the new proteins that are synthesized by the ribosomes. Continuous with the rough endoplasmic reticulum is the smooth endoplasmic reticulum. The smooth endoplasmic reticulum does not have any ribosomes, and hence it does not play a part in protein synthesis. Instead, it plays a key role in the synthesis and metabolism of phospholipids and cholesterol. It absorbs and transports lipids obtained from the gastrointestinal tract, and it detoxifies substances such as pesticides using enzymes. It also acts as a reservoir for calcium ions, storing and releasing calcium during muscle contractions. The Golgi apparatus consists of stacked and flattened membranous sacs. These membranes can form enclosed spherical containers called vesicles. Proteins and lipids made in the endoplasmic reticulum are modified, concentrated, and packaged into vesicles and released from the cell through a process called exocytosis. The mitochondria is one of the most essential organelles within a cell. A mitochondria is enclosed by two membranes. The outer membrane is a smooth and featureless membrane, whilst the inner membrane is folded inwards, forming a shelf-like structure called the cristae. The cristae protrudes into a gel-like substance called the mitochondrial matrix. One of the most interesting features of a mitochondria is that they have their own DNA and RNA, and are also capable of manufacturing their own ribosome. Mitochondria are known as the powerhouse of the cell because they produce ATP, which is the energy source of the cell, by burning glucose in the presence of oxygen. Using glucose in the presence of oxygen, mitochondria can produce up to 38 molecules of ATP. However, in the absence of oxygen, they can only produce 2 molecules of ATP. Lysosomes are single membrane-bound structures which contain up to 40 different activated digestive enzymes. Lysosomes are very common in phagocytes, since they help digest particles that the cell has engulfed, such as bacteria, viruses, or toxins. Furthermore, they can also dispose of stressed, non-functional, or dead cells and organelles. When lysosomes rupture within cells to digest themselves via the release of various digestive enzymes, this process is called autolysis. Peroxisomes are spherical membranous sacs containing a variety of enzymes. The enzymes within peroxisomes neutralize highly reactive chemicals, such as oxygen-free radicals, by converting them to hydrogen peroxide, and then eventually degrading them to oxygen and water. Peroxisomes are especially numerous in the cells of the liver and kidney because of their role in detoxification. The extracellular matrix is the unstructured material secreted by the cells that exists between the cells. It contains adhesion molecules that hold and adhere cells to the connective tissue fibers, as well as proteoglycans that are known to regulate the movement of similar molecules such as sodium, potassium, and water. In this particular slide, we will try and understand the relationship between different organelles and the cellular transport that occurs within the endomembrane system of a cell. The endomembrane system is comprised of different organelles that are involved in the production, storage, and export of various proteins, lipids, and other molecules. For this to work, several organelles have to work synergistically. Whenever there is a need to produce a protein, mRNA and ribosomes are synthesized inside the nucleus and are then exported to the rough endoplasmic reticulum, or the cytoplasm, via the nuclear pores. Once the mRNA enters into the rough endoplasmic reticulum, where proteins are synthesized by ribosomes, these proteins are then moved to the Golgi Apparatus, where proteins ready for exportation are enclosed within the vesicles and sent to the plasma membrane. Here, the vesicular membrane fuses with the plasma membrane, and the proteins are released from the cell through a process called exocytosis. Proteins that are needed in the nucleus will be transported into the nucleus through the nuclear pores. Some other vesicles produced by the Golgi apparatus give rise to the lysosomes and vacuoles. In summary, from this video, we have learnt about the key differences between prokaryotes and eukaryotes. We've also learnt about the structure and function of the plasma membrane and all the other organelles of a eukaryotic cell. And we have learnt the role of the extracellular matrix and how cellular transport works.